I want to welcome everybody to this month's Centers of Excellence in Hematology Meet the Experts series. If you haven't joined us before, welcome. If you have, welcome again. And today we're pleased to have Dr. Anna Beaudin from the University of Utah speak to us. Before she speaks to us, I'm going to share my screen for just a minute to let you look at and guide you through the website for this center. Can you guys see that and my cursor? Yes? Yes. Okay, I can't see anybody else but you. Sorry. Okay, so this is the website for the Centers of Excellence in Hematology. And there are lots of links here that you can go through. You can find out about different centers that are available here. If you do this drop-down menu, there's one at Indiana, one at the Fred Hutch, one at Cincinnati, one at the University of Utah, and one at Yale. And they have different core facilities at each of those that I encourage you to go look at to see if there's a course that you could be using. To that end, if you go to this funding link right here, you will find that we offer opportunities for funds. And right now there are two types of opportunities. There are what are called type A P&F funds. And those are essentially core credits where you apply, it's like a one-page application to utilize a core somewhere. And these you can apply for anytime. You could send it today, you can send it in 5 weeks, whatever, 6 months. These are always available and it's a very simple process. The second type are called type B awards, and they're up to $75,000 for 1 year. They're really focused on pilot research projects that generate preliminary data for larger research grants. And the application deadline was last March 15th. So right now we are not taking type B grants, but if you keep your eye on this website, there will be another call in a couple of months. So I highly recommend you go visit this site. And now I'm going to stop my sharing. And then I want to introduce Dr. Beaudin to you. So Dr. Beaudin's training is highly interdisciplinary, which she's got biochemistry and nutritional science and developmental biology in her wheelhouse. And she originally was at UCLA, where she completed a bachelor's degree in psychology. Is that UCLA? Is that right?
No, I was at Cornell for
I beg your pardon. I thought it was originally from LA. Then you went and did your psychology degree and a Ph.D. in nutritional science at Cornell University. She then went on to a postdoctoral fellowship at UC Santa Cruz, one of my favorite places to go surf, where she used fate mapping approach to discover novel transient hematopoietic progenitors that serve as a cell of origin for fetal restricted innate-like lymphocytes. And she opened a lab in California first, but has since come to Utah. She's well-funded, NIH funded. She's a recipient of the Pew Biomedical Scholars Award, and she's an active member in a lot of hematology societies, as well as an active reviewer for many high-tiered journals. And I want to turn the time over to her, where I'll let her share her screen and introduce the title of her talk today. And thank you all for joining us. For the presentation, I'm going to ask everybody to mute themselves except for Dr. Beaudin. And then if you have questions, you can populate them in the chat and I will invite you to unmute to ask your questions. And Dr. Beaudin has agreed to allow sort of that interruption piece that we do because it's more fun. And thank you again for speaking today.
Of course. Thank you, Diane, for the kind introduction. And please feel free to interrupt me as I go. I'd much rather answer questions as they arise. So I'm super pleased to discuss my work today, some of the work in my lab today, looking at the effects of prenatal inflammation on developmental hematopoiesis. Excuse me. Okay, so the overarching question that my lab seeks to address is how perturbation of immune or hematopoietic development impacts health and disease susceptibility across the lifespan. And so we all know that across our lifespan, our risk of disease increases. And we now know that for most of these age-related diseases, chronic inflammation is a major driver of age-related disease. And while for most diseases, there's some fixed genetic contribution to disease risk, in the case of age-related disease and inflammation, this fixed genetic contribution is really relatively nebulous. Instead, we know from accumulating epidemiological evidence that adverse events during early life are cumulatively associated with a greater risk for this type of age-related and inflammation-associated disease than genetic risk alone. Now, we often don't intervene for these types of diseases until adulthood when there's already a significant onset of symptoms. And this type of intervention in adulthood is associated with only a modest reduction in risk from disease. So our approach is to try to understand how these adverse events during early life drive risk for disease across the lifespan. And the idea is to try to identify time points and approaches for intervention much earlier in the lifespan, and therefore have a much greater impact in mitigating disease susceptibility across the lifespan. And we approach this from the perspective of hematopoiesis, and particularly from the perspective of developmental hematopoiesis, which is a very dynamic process that occurs across many anatomical organs during development. And so kind of the classic dogma of developmental hematopoiesis is that it occurs in waves of hematopoietic cell generation. And the classic, I guess the classic dogma, I already said that, but the classic dogma is that it occurs in kind of two primary waves. The initial wave is referred to as primitive hematopoiesis, and this initiates in the very early developing embryo in the extraembryonic yolk sac. And this initial primitive wave of hematopoiesis generates a very limited subset of blood cells, primarily primitive red blood cells and some megakaryocytes and macrophages. And this was really initially thought to just serve the early oxygenation and scavenging needs of the developing embryo. Subsequently, in the mid-gestation embryo, this is in the mouse, but a fairly similar trajectory occurs in humans. Subsequently, in the mid-gestation mouse embryo, we get the generation of the first definitive hematopoietic stem cells that generate definitive hematopoiesis. And these definitive hematopoietic stem cells are now characterized by their ability to generate all mature blood cells, including definitive red blood cells, as well as granulocyte macrophages, and then also cells of your adaptive immune system like B cells and T cells. These definitive hematopoietic stem cells arise from hemogenic endothelium in the developing aorta. We call this the AGM, or aorta gonad mesonephros region. And these definitive hematopoietic stem cells that arise in the mid-gestation embryo are capable, when isolated from the embryo, of regenerating the adult blood system upon transplantation. So they can reconstitute the adult blood system. And it is on this basis that we've generally considered these definitive HSCs that arise in the developing embryo to be the precursors of the adult hematopoietic stem cell system. Oh, sorry. And so these definitive HSCs, once they bud out of the developing aorta, they hit the circulation and they seed the fetal liver, which is the primary hematopoietic organ during development, and where they undergo massive expansion. And then right around birth, they move from the fetal liver to the bone marrow, where again, they're thought to give rise ultimately to the adult hematopoietic stem cell compartment in the bone marrow. Now, we've generally tended to think of this process of hematopoiesis is fairly linear. And similarly, the kind of the dogma has been that immune development is also fairly linear, that as these definitive HSCs that give rise to all mature blood and immune cells mature across development, so do their immune cell progeny. Now, this picture has really changed over the last decade or so with the advent of more sophisticated genetic approaches like lineage tracing, and as well as like things like lineage barcoding. We now know that this picture is far more complicated. So we now know that fetal hematopoiesis is really underlaid by many overlapping waves of hematopoietic cell generation that are driven by distinct hematopoietic progenitors. And part of my lab's interest is really to understand how does this heterogeneity that makes up fetal hematopoiesis ultimately contribute to the heterogeneity that we now know exists in the adult bone marrow. So we now know that there's no such thing as one hematopoietic stem cell. The adult hematopoietic stem cell compartment is very heterogeneous. And we know that this heterogeneity in adult hematopoietic stem cell compartment really underlies many important parts of the adult immune system. So we know that underlies obviously differences in the trajectory of the immune output in the adults. As hematopoietic stem cells give rise to all immune cells, obviously heterogeneity in your adult HSC compartment will change the trajectory of your immune output. We also know that HSC heterogeneity underlies a differential response to inflammation, as it's been shown that different types of HSCs can differentially respond to inflammatory cues. And finally, we believe that HSC heterogeneity might underlie propensities for clonal expansion, which we know predisposes individuals to age-related diseases such as leukemia, cardiovascular disease, and cerebrovascular disease. Okay, so the other thing that we know from parallel types of approaches, including fate mapping within the field of immunology, is that these waves of hematopoietic cell generation during fetal development also give rise to very distinct subsets of immune cells during fetal life. And I'm showing you a few examples of these types of cells that are produced during fetal life. So these kind of come in two non-mutually exclusive and overlapping flavors that include tissue resident cells and also cells that fall into a category that we refer to as innate-like lymphocytes. So in terms of tissue resident cells, these include cells like tissue resident macrophages, like microglia in the brain, Langerhans cells in the skin, Kupffer cells, and subsets of alveolar macrophages in the lung and cardiac macrophages. And these also include cells like that we refer to as innate-like lymphocytes, that are cells that look like conventional lymphocytes, but they actually function kind of at the border of innate and adaptive immunity. These cells are quite functionally distinct from similar cells that might be made during adult bone marrow hematopoiesis. As I mentioned, in the case of innate-like lymphocytes, these are cells that really don't fit into either adaptive or innate immunity. They're more like innate immune cells, even though they look like lymphocytes. In the case of tissue-resident cells, tissue-resident macrophages, these are cells that function both in tissue immunity, but also in tissue homeostasis. And often these are tissue resident cells that seed the tissues as they're developing, and they acquire very tissue specific functions. Importantly, these cells show very poor or limited regeneration from adult bone marrow hematopoiesis. And in cases where it's been shown that these cells can be forced to be made from adult bone marrow HSCs, it's unclear whether these cells are ultimately the same cells as those made during development. So this is very important because these cells are made once during fetal life, and they persist in their tissues of residence or for the lifespan of the organism, and they show very poor regeneration for adult bone marrow. Because of this, these cells have a unique capacity to self-renew or self-maintain within their tissues of residence, and this capacity is still very poorly understood. Additionally, despite the fact that we now know that these cells are made from fetal precursors, the developmental mechanisms that specify these cells are also very poorly understood. And part of my lab's work is to understand at a very basic level how fetal hematopoiesis produces immune cells that are so functionally different than the cells produced from adult hematopoiesis. But one of the things I want to focus on for the purpose of this talk is that many of these cells have more recently been implicated as being dysregulated in tissue-specific diseases and in diseases of tolerance. And so we became interested in how do these cells become dysregulated very, very early, and how does this predispose individuals to disease susceptibility? And so one of the ways that I became interested in this problem was based on some of the work I did in my postdoc at UC Santa Cruz, where we used a fate mapping approach to define the contribution of fetal hematopoiesis to adult immunity. And in this work that was published several years ago in Cell Stem Cell, we used a model that we refer to as the flick switch mouse to look at fetal hematopoietic differentiation pathways. And in this model, which I won't go into too much detail for because the model itself in terms of a fate mapping approach is not that important for the purposes of the story. But we used a mouse where we used a model in which we crossed mice expressing Cre recombinase off of the FLIC2 or FLIC3 promoter to the MTMG reporter, in which you have a tomato, mice expressing tomato with a stop lox and then a GFP reporter. And so in these mice, All cells start off expressing tomato, and then when they express FLIC2, they undergo an irreversible switch where the tomato coding region is floxed out, and you get an irreversible switch from tomato to GFP expression. And what was originally reported in this model is that all adult HSCs in this model remained tomato positive. And what that means is that any cell that had switched to GFP during development could not contribute to the adult HSC compartment because that's an irreversible genetic switch. So all cells, all adult HSCs in this model had never had a history of FLIC2 or FLIC3 expression. And so when I started looking at this model during development, I found indeed that there were tomato positive HSCs during development that, as the classic model goes, seeded the neonate bone marrow and seeded the adult HSC compartment. And we referred to these tomato HSCs that we found in the fetal liver as our classic adult HSCs. But we also found a GFP positive HSC in the fetal liver. And we found that this GFP positive fetal liver HSC, like its tomato positive counterpart, was capable upon transplantation of reconstituting the adult blood system. So it acted like a bona fide HSC upon transplantation. Nevertheless, using this irreversible fate mapping model, we found that despite a limited capability to seed the bone marrow, it did not persist into the adult bone marrow and contribute to the adult HSC compartment. And so we refer to this HSC as a developmentally restricted HSC or a DRHSC. And we further went on to show that this GFP positive DRHSC was functionally distinct from its tomato positive counterpart, and specifically that it was a lymphoid biased HSC, both in terms of output and in terms and that it was transcriptionally lymphoid biased. And we also showed that it had a specific capability to generate some of these fetal restricted innate-like lymphocytes, including B1 B cells and V gamma 3 expressing T cells. And so what this really defined for us, what this really conceptualized for us was that we had identified a specific transient progenitor in the fetal liver that was responsible for the generation of fetal restricted immune cells that go on to persist into the postnatal period and contribute to the adult immune system. And this really conceptualized for us what we referred to as a critical developmental window, wherein any extrinsic or intrinsic inputs during this window now shape the phenotype of the adult organism, or in this case, the adult immune system. And so we decided to test this idea of the critical window by applying extrinsic inputs during this window, including things like infection or inflammation, maternal alterations in maternal diet or maternal exposure, and test how this affects the establishment of fetal hematopoietic stem cells, the output and function of these fetal-derived immune cells, how this ultimately shapes the adult hematopoietic stem cell compartment, and how all of these changes ultimately impinge upon immune function in adulthood and risk for disease. And so some of the work I want to start with today is to tell you how we started to do this work as kind of a proof of principle of this using a maternal infection and inflammation model. And this is work performed by two very talented graduate students in my lab, April Apostle, whose defense date was just set for 3 weeks from now, and Diego Lopez, who is still currently in my lab at University of Utah. So when I first started this work, we really didn't have access to any infection models. So we turned to kind of a model of maternal infection that is used in the neuroliterature, referred to as maternal immune activation. And here what we're doing is we're simply mimicking a mild viral infection in a pregnant mom by injecting a polyice, a TLR3 agonist at E14.5, which is the point at which fetal liver HSCs have maximally expanded within the fetal liver. And so the first thing we observed was that there was a significant expansion of the fetal hematopoietic stem and progenitor compartment, or LSK, within the fetal liver. So again, we're just injecting the mom and we're looking at hematopoiesis in the fetus. And so we wanted to look at what was happening within this expanded hematopoietic stem and progenitor compartment. And so one of the things we wanted to look at was the differential effects on HSCs and various multipotent progenitors. And so this has been really well established or really well investigated within the adult hematopoietic system, where we know that these various MPPs are The differentiation of MPPs from HSCs is balanced to provide balanced lineage output downstream of the HSPC compartment among lymphoid and myeloid and MEGI output. And this is a nice review by Eric Petras, a friend of mine. And we also know that in response to a wide variety of inflammatory stimuli, be it different cytokines, infections, TLR agonists, et cetera, we know that in response to a wide variety of inflammatory stimuli in the adult, what you uniformly observe is myeloid bias. And this is accomplished both by the activation of myeloid biased hematopoietic stem cells, as well as by the downstream expansion of myeloid biased multipotent progenitors. So when we invoked prenatal inflammation during fetal hematopoiesis, what we observed instead was the immediate expansion of our lymphoid biased GFP positive DRHSC. So this is 1 day following injection of PIC into the mom. And immediately downstream of that, what we observed was expansion of our lymphoid biased multipotent progenitor. So we wanted to test if downstream expansion of our lymphoid biased multipotent progenitor occurred downstream of our GFP positive DRHSC expansion. And so what we did to test this was to inject poly-IC into the mom, sort out our 2 HSC populations, tomato positive HSC and GFP positive DRHSC, and stick them in culture for 24 hours and see what happened. And so what we observed is that our GFP positive DRHSC, even under our saline conditions, already produced more MPP4s than our tomato positive HSC. So this was consistent with lymphoid bias we documented both upon transplantation and at the transcriptional level in our previous publication. But when we induced prenatal inflammation in situ with poly-IC, what we observed was that this bias was further exaggerated. So now our GFP positive DRHSC was pumping out MPP4s, which is our lymphoid bias multipotent progenitor subset, at the expense of other MPPs, suggesting that this downstream expansion of lymphoid bias MPP4s in situ was occurring because of our expansion of our GFP positive DRHSC4. So in this case, as opposed to what we see in the adult, prenatal inflammation was specifically invoking a lymphoid bias in situ. We next wanted to test how prenatal inflammation affected fetal HSC function in isolation. And so we performed transplantation assays by pulling out our tomato positive, GFP positive, and GFP positive HSCs and performing transplantation in competition with 5 times 10 to the 5th adult whole bone marrow cells into lethally irradiated mice. And we did this both in a primary and a secondary transplant.
Hey, Anna, can I ask a question that's back? If you have this bias for lymphoid, do the animals that you transplant into develop an anemia? Do they have a problem making red cells?
No, so they're still, these are still multipotent stem cells. So they still make all lineages. They just make more lymphoid cells. So if you compare the output of myeloid to lymphoid, they make more lymphoid as compared to myeloid. Okay.
Thanks.
No problem. So we pull these cells out of the fetal liver and we transplant them into irradiated adult recipients. And these data look very similar on the left in our saline condition to what we've initially reported in our paper, in our 2016 paper, which is that within the phenotypic fraction that we transplant, there are less GFP positive HSCs than tomato positive HSCs. Nonetheless, poly-IC really doesn't affect LTMR in a primary transplant. And when we look at the readout of these cells over 16 weeks in the peripheral blood, we can see that the tomato positive HSC, when hit by maternal immune activation or prenatal inflammation, we do see that we do see a slight myeloid bias readout in the peripheral blood. So we see that these cells are generating more granulocyte macrophages upon induction with prenatal inflammation. But otherwise, we don't see any really severe dramatic changes. And then with our GFP positive DRHSC, we don't see any extreme cellular intrinsic changes that induced by prenatal inflammation. In other words, these cells we don't see any more lymphoid output that is induced by prenatal inflammation. And this is again, when these cells are isolated from the fetal liver and put into an adult recipient. Now,
we have a question in the chat from Shelby Steinmeier. Shelby, do you want to unmute and ask your question?
Yeah, sure. So I was just wondering if you have looked in fetal bone marrow and seen if this lymphoid bias persists in stem cells from the bone marrow compared to the fetal liver.
So we have looked to see that these cells are in the bone marrow. They seed the bone marrow in lower numbers, but we have not transplanted them from the bone marrow. We have mostly focused on their existence within the fetal liver because there are less of them as you move into the bone marrow. They decrease in number. And we think, so they're also more proliferative at baseline. And so we think they just get outcompeted once they see the bone marrow.
Thank you.
That's a good question. Thank you. Okay. So there's some differences here, and we do see that prenatal inflammation induces a myeloid bias in our tomato-positive HSCs upon transplantation. But when we move into the secondary transplant setting, we see a very different story where now our tomato-positive HSCs show a significant reduction in the number of recipients that show long-term multilineage reconstitution. Our GFP-positive DRHSCs see a slight reduction, but it's not severe. And then when we look at readout in our secondary transplants, we see this extremely robustly, where our tomato-positive HSCs that were initially exposed to prenatal inflammation show extreme exhaustion phenotype in a secondary transplant, whereas our GFP-positive DRHSCs do not. So this is a very disparate response among these two heterogeneous fetal HSC populations, where our tomato-positive HSCs are looking like adult HSCs. This is what adult HSCs do in response to inflammation. They invoke a myeloid bias and they exhaust, whereas our GFP-positive DRHSCs are not doing that. They are not functioning. They don't evoke a lymphoid bias as they do in situ in the fetal liver, but they also are not responding like an adult HSC, invoking a myeloid bias and exhausting upon secondary transplantation. So this indicates a fundamentally different response between these two HSC populations. So we next, as some people do, turn to single-seq to look at some of the molecular changes that were underlying this in vivo response to inflammation. And in this case, we sequenced over 24,000 cells in saline and PIC conditions. And we got 14 different clusters this is just within the hematopoietic stem and progenitor compartment isolated from the fetal liver 1 day after maternal immune activation. And one of the first things we did with these 14 clusters was to take bulk sequencing profiles of our tomato-positive HSC and our GFP-positive DRHSC and to overlay these profiles onto our clusters. And as you can see, these two profiles completely bifurcate on these 14 clusters. So our tomato-positive HSC profile kind of overlays on these clusters on the right, clusters 2, 7, 4, and our GFP-positive DRHSC overlays on these clusters on the left. And when we look at how these clusters change in response to PIC, we can see that the clusters that overlay with the tomato-positive HSC are decreasing in response to PIC, whereas our clusters that overlay with our DFP-positive DRHSC on the left are increasing in response to poly-IC. So this is similar to what we have seen in situ. Now we wanted to identify which of these clusters really was more closely associated with our actual HSC. And so we also took these clusters that we identified in single cell seq and overlaid them with adult HSC and MPP profiles. And so what we found here were that there were two clusters that were both overlapping with HSC profiles and non-overlapping with MPP profiles. And these corresponded to cluster 2 for the tomato positive HSC and cluster 9 for the GFP positive DRHSC. And when we performed GSEA with our tomato positive HSC and our GFP positive DRHSC identities, we found that these were highly significant, cluster 2 for tomato and cluster 9 for GFP positive DRHSC. So even though we found multiple clusters that overlaid with these kind of bulk sequencing profiles that were not as pure as our single cell sequencing can reveal, we kind of felt comfortable assigning cluster 9 as our DRHSC and cluster 2 as our tomato positive HSC. So taking these identities, we kind of then moved to something different. We wanted to try to understand what was evoking this differential response between these two different HSCs. And we went and profiled cytokines at multiple levels in maternal serum, in the amniotic fluid, and within the fetal liver in response to our prenatal inflammation invoked by MIA. And here I'm showing you just a cytokine profile within fetal liver supernatant. And so what you can see here is that only 2 cytokines were upregulated in response to MIA induced with poly-IC, IL-1 alpha and not surprisingly interferon alpha. And so what we then did was to kind of leverage this data back with our single cell sequencing to try to determine which of these clusters is ultimately responding to one of these 2 signals. And to make a long story short for IL-1 alpha, we found that none of these clusters responded to IL-1 alpha. In fact, there is very little IL-1 alpha receptor or IL-1 receptor signaling components present within the fetal HSPC compartment. Instead, what we found is that type 1 interferon signaling was highly enriched within our cluster 9, which is the cluster we found associated with our GFP positive DRHSC. And when we went downstream and looked at cells associated with IFNAR signaling, we found that these genes were highly upregulated within clusters overlapping with our GFP positive DRHSC, or highly enriched, in clusters associated with our GFP positive DRHSC. And that when we induced inflammation with PIC, that these genes were also upregulated within those clusters. We also wanted to test how this translated directly into the response of our HSPCs. And so we used MX1 Cree model crossed to the AI9 reporter as kind of a lineage trace of the response to type 1 interferons. And so what we did was to cross these 2 mice and then inject the mom with poly-IC to read out the IFNAR signaling response in the fetal liver a 1 hour, 24 hour, or 72 hours later. And so this is just within the HSPC compartment, we're tracking the response of TD tomato positive cells within the fetal liver. And what you can see, this is unfortunately, we don't have these mice also crossed into our flick switch model, so we can't differentiate our tomato positive and our GFP positive DRHSC. But our DRHSC is kind of caught in between our long-term HSC and our short-term HSC designation. It's kind of more within our short-term HSC designation than within our long-term HSC designation. But you can see how rapidly this signal expands between a 1 hour and a 72 hour. And we also see a smaller but significant expansion within our MPP4s. So this suggests, all of these data together suggests that our GFP positive DRHSC is really responding to this type 1 interferons that are upregulated in the fetal liver by MIA induced in the mom. And this drives a downstream cascade to our MPP4s. And this is exactly both what we see in vivo and in our single cell sequencing. So type 1 interferons specifically activate both phenotypically and transcriptionally identified DRHSCs. So coming back around to this idea of the critical window, we wanted to look at how these types of changes we observed induced by prenatal inflammation and fetal hematopoiesis translated into changes into hematopoiesis and immune function postnatally. And so we first looked in the bone marrow in postnatal day 14 hematopoiesis. And we saw that our hematopoietic stem and progenitor compartment was still expanded. So this is 3 weeks after intervention and 2 weeks after birth. And we saw that there were no changes to conventional long-term HSCs or to our tomato positive HSCs, but there was a significant increase still in our GFP positive DRHSCs in the bone marrow, as well as lymphoid bias FLIC2 positive MPP4s. So this was still sustained 3 weeks post MIA induction. When we looked in the adult, we similarly saw quantitative changes to the adult bone marrow. By frequency, HSPCs were still increased. We similarly saw increases, sustained increases to the GFP positive DRHSC and to FLIC2 positive MPP4s. And we really think that this was driven by a sustained increase in cellularity of the GFP positive DRHSC into adulthood. Similarly, as I mentioned before, we have previously shown that this DRHSC has a specific capability to generate these fetal-derived innate-like lymphocytes, including B1A and B1B cells in the peritoneal cavity, as well as marginal zone cells in the spleen. And so we wanted to see if the inappropriate expansion of this GFP positive DRHSC into the postnatal period was associated with the expansion of its innate-like lymphocyte progeny. And so here I'm showing you the quantification of those cells, both in the peritoneal cavity and in the spleen. And what we observed was the specific expansion of these innate-like lymphocyte subsets, including B1A and B1B cells in the peritoneal cavity, as well as marginal zone cells in the spleen, but not conventional B2 cells or follicular zone cells within the spleen. We also wanted to test how this prenatal inflammation and activation specifically of the GFP positive DRHSC affected the function of these innate-like lymphocytes. And so we looked at specifically the output of these innate-like lymphocytes, which is the production of natural antibody like IgM and IgG3. And we observed that not only was the concentration of these natural antibodies like IgM and IgG3 elevated within the peritoneal cavity, but this is controlled for the increase in the number of cells within the peritoneal cavity. So this is concentration controlled on a per cell basis. So not only are these cells increased on, not only is the cellularity of this compartment expanded, but these cells are also hyperactivated. So they're pumping out natural antibody on a per cell basis. We also tested their capability to produce IL-10 upon stimulation with PMA and LPS. And what we observed is that these cells are also producing more IL-10 on a per cell basis. So these cells are not only expanded, but they're also hyperactivated in response to prenatal inflammation. So far, we believe these data provide a proof of principle that prenatal inflammation defines the framework for the idea of a critical window of hematopoietic and immune development, where an extrinsic input such as prenatal inflammation affects the establishment of fetal hematopoiesis by driving lymphoid bias in the expansion of lymphoid biased hematopoietic stem and progenitors. This ends up driving the expansion and hyperactivation of fetal-derived innate-like lymphocyte progeny. And finally, this ultimately might result in persistent remodeling of the adult hematopoietic stem and progenitor compartment. And we're still very interested in testing how this ultimately remodels an adult immune output and risk for disease. And so I was just checking the time. I think I still have a few more minutes. I'd like to quickly show you how we are now moving into this second question and how this modifies disease risk. And so this is what we joke is Diego's second PhD project, where he's now looking at how prenatal inflammation shapes the output and function of another type of innate-like lymphoid cell. And this is the innate lymphoid cell type 2 or ILC2, which is also a developmentally layered cell that has been recently shown by Rich Loxley's lab to at least in part be produced during fetal development. So what Diego has asked is how prenatal inflammation shapes risk for asthma susceptibility by altering fetal ILC progenitor establishment and ultimately remodeling lung immune landscape and function by shaping the output and function of ILC2s, which are one of the earliest cells to seed the developing lung. And so he's used the exact same model here, maternal immune activation with poly-IC, and asked how this shapes function and output of ILC2s in the neonate lung. And one of the first pieces of data he collected in my lab while we were still at UC Merced was to show that this also changes the output and function, of course, of lung ILC2s. So he showed that these cells were not only expanded in the lung by several fold, in the neonate, But he also showed that these were hyperactivated cells. So these are cells that function kind of like Th2 cells before Th2 cells exist in the lung. So these are again cells that look like lymphocytes, but they don't have specific receptors, but they pump out Th2 type. They produce Th2 cytokines before they are Th2 cells there. Th2 cells there, excuse me. So these cells produce IL-5 and IL-13. And what I'm showing you here is that when these cells have been, when these cells come off, are produced in offspring that have been exposed to prenatal inflammation induced by poly-IC, these cells pump out these type 2 cytokines like IL-5 and IL-13 in the absence of any other stimulus. So these are just cells that have been pulled out of the lung and hit with PMA and ionomycin. These have not been stimulated in any other sense. He also showed that these expanded and hyperactivated ILC2s in the lungs also have an inflammatory phenotype as determined by KLRG1 expression. So these are different cells coming off of a mouse that has been exposed to prenatal inflammation. So he wanted to test what happens to the progenitors of these cells, because these ILC2s don't exist at the time that we've induced prenatal inflammation in the fetus. And so he looked at progenitors as early as E15-5, so this is 1 day after induction of prenatal inflammation, and he saw that these cells are already increased by cellularity and frequency, and that this increase is maintained across fetal development in the lung, in the liver. He then looked at how these cells proliferate and expand postnatally, and he observed that even immediately after birth, these ILC2s in the lung are already proliferating more than their saline counterparts in response to prenatal treatment with poly-IC. And this really drives ultimately the increase that we see at postnatal day 14. And I should say that we picked postnatal day 14 as the time point to initially examine, because that's the peak of cellularity of ILC2s in the lung across the lifespan. After that, these cells normally decline, and then they're replaced actually by Th2 cells. So we see this increase in these ILC2 cells and this hyperproduction of Th2 cytokines early in lung development. And as I mentioned, these are some of the earliest cells to seed the lung. And so we wanted to understand how this impacted the lung immune landscape and lung immune development. And so Diego also profiled all these other cell types in the lung at P14. And what he saw was that the lung immune landscape was completely remodeled. So we see significant increases in a number of different lung immune cell types, including B cells and T cells, NK cells, NKT cells, alveolar macrophages, eosinophils, et cetera. And this probably isn't that surprising, because these cytokines are meant to recruit cells in the face of, IL-5 and IL-13. These are cytokines that are intended to recruit. So one of the things Diego wanted to do was to see, well, are these, we hypothesized that these expanded and hyperactivated ILC2s were the cells that were bringing in all these other cell types, but we wanted to test that. And one of the initial ways he tested that was to just profile these changes over time and see, were these other cell types coming in before the ILC2s, or were the ILC2s expanding before any of these other cell types? And sure enough, what we observed was that our ILC2s were significantly expanded and proliferating before any of these other cell types were changing. So we really think it's the expansion and hyperactivation of these ILC2s that is driving the change in phenotype for all these other cell types.
Hey, Anna, I have a question. You talked about how normally the ISC2s will diminish over time. When you have, is there a difference in the lifetime of those cells if you do inflammation or not?
Great question. So we're now performing experiments where we are fate mapping the cells that get hit by prenatal inflammation. So Diego, I won't show these data today because they're still in progress, but so Diego has done two things. He's tracked out the persistence of this expanded phenotype, and he has shown that all ILC2s contract after P14, but even though the population generally contracts in response to prenatal inflammation, that population is still expanded relative to saline controls. But we want to know specifically what happens to the cells that are hit during development. And so he's using an ID2 Cree ERT2 to fate map those specific cells out into adulthood. Does that make sense? Did I answer your question?
You did. Thanks.
Okay, great. Okay. So one of the other critical questions we wanted to ask was, are these changes being caused because the ILC2s are changed? Is this cell intrinsic, or are we damaging something within the lung or perturbing? Because these ILC2s also respond to alarmins in the lung. If the lung is perturbed, then that'll hit the ILC2s and perturb their function. And so Diego decided to profile the lung cytokine and immune environment to test if any of these alarmins were up, and then thereby altering ILC2 function. And so he did this with the help of an undergraduate here at University of Utah, Leah Griffin. And what he found was that there were no alterations to any of the alarmins that would have set us off to think, oh, the lung microenvironment is perturbed, and that is what is perturbing ILC2s. Instead, even by P9, he already sees significant difference in IL-5 detectable within the lung within our MIA treated offspring. So this suggests that this is actually a cell intrinsic defect or alteration induced by prenatal inflammation that is again driving these changes to the lung immune landscape. So one of the last pieces of data I want to show you today is that we are again testing for disease phenotype here. And one of the hardest things that we've had to set up in the lab is these house dust mite challenges. So we're actually trying to test for asthma susceptibility in these neonate mice that have been exposed prenatally to inflammation induced by poly-IC. So here what Diego set up was that he was challenging these mice 5 times a week for 4 weeks with house dust mite given intranasally. And then, unfortunately, with black 6 mice, it's quite challenging to measure airway hyperresponsiveness. We actually have now at Utah some fantastic collaborators that are helping us to overcome some of those challenges. But we were told previously, for example, by the asthma core at UCSF that black 6 mice were not capable of measuring airway hyperresponsiveness of black 6 mice. So instead, so far, what we've done is to use pathology to tell us whether mice have asthma. And so here what I'm showing you are sections of the lung stained with hematoxin eosin and also a periodic acid shift stain for mucus. And what you can see here is that normally both the vasculature and the airways are nice and open, but when you start treating with house dust mite, what you get is immune cell infiltration that blocks this off. And what you can see is that when the mice have been treated during fetal development with poly-IC that induces inflammation, that both airways and vasculature are completely blocked off by this immune cell infiltration. And we can see that also by our FACS analysis.
We have a question in the chat. Sorry to interrupt.
No.
Let's see. The question is from Hitish. I'm going to mess this up, but Deshmukh, would you like to unmute and ask your question? I'm not hearing. Perhaps I can read it and then you can respond.
Sure.
It says, does prenatal inflammation remodel the ILC compartment in extrapulmonary sites, for example, the gut? Fetal inflammation is a risk factor for necrotizing enterocolitis, a fatal inflammatory condition in preterm infants.
Yeah, it's a great question. And I will say that every time I give this talk, I get asked about a different ILC subset in a different tissue. I imagine that it does. We did start some work looking at different ILC subsets in the gut, but the truth is that I'm not a gut person, and so we haven't moved forward with that work. But I mean, the principal idea here is that we are hitting progenitors, and these are progenitors for all subsets. I mean, we're starting at the stem cell and moving down. And so I would expect that this would influence subsets that move into the gut and to move into many different peripheral tissues. But we can only investigate one subset at a time. That's the best answer I have. That's a great question.
Thanks.
Yeah, thank you. So to conclude on this, this data, these are studies that are still ongoing, but we have some good, strong preliminary data to suggest that this persistent and hyperactivated ILC2 phenotype is really driving sensitivity to house dust mite induced airway inflammation. So to conclude the work that I've shown you today, we really believe that our work defines what we refer to as a framework for a critical window of hematopoietic and immune development, wherein hematopoietic and immune development can be shaped by extrinsic inputs during development, including prenatal inflammation. And that this shapes both the output and the function of fetal-derived immune cells from the progenitor. And that we've shown that prenatal inflammation activates transient lymphoid biased progenitors as opposed to what we see in adulthood. And that we think this functions to both shape the postnatal immune response and also spare adult HSCs during fetal life. We think that both the maternal immune response to inflammation and also the fetal immune response to inflammation act as drivers as ultimately of this fetal hematopoietic and immune response. I didn't have a chance to talk a lot about that today, but we have done some work with another infection model, Toxoplasma gondii, congenital toxoplasmosis, where we've done some work to try to dissect the maternal versus the fetal immune response to inflammation. I hope to get another chance to talk about that work another time. And finally, I hope I've had a chance to show you that we believe this work has that prenatal inflammation and the effects of prenatal inflammation on both fetal hematopoiesis and fetal immune development has long-term consequences for both normal and abnormal immune function. And with that, I'd like to take any additional questions and to thank you all for listening, to thank the people that have really done this work, April Apostle, and particularly Diego Lopez, as well as some other talented people in my lab, Eric Liebisch and Clint Valencia, who's actually, both Eric and Clint are also at Utah. Eric is a technician in another lab, and Clint Valencia is a second-year graduate student in another lab here at HCI. To thank my funding sources and just to also mention that I'm also recruiting postdocs. If anybody is interested, please feel free to reach out and contact me. Thank you so much.
Thank you, Anna. I want to invite anybody else who has questions to unmute themselves and go ahead and ask it. We'll just give that a minute to see if anything pops up.
Sure.
So as you might expect, yeah, go ahead, please.
Oh, sorry. So it sounds like you've looked at toxoplasma as a kind of another type of fetal inflammation. And I'm just wondering if there are like other models that you've thought of and like kind of maybe like trying to like recreate like chorioamnionitis or something like that as a fetal exposure.
Yeah, so I mean, modeling congenital infections in mice is not for the faint at heart. That's why we started. I mean, I started with poly-IC in part because when I initiated this work at the end of my postdoc, I was in an institution that had no immunology, so there was no opportunity to do infection models. But it turns out that poly-IC is a really nice model to tease things apart with. And with the toxo model, one of the things that's been nice with that is that we've done some nice work comparing, you know, a full-blown infection with injections of single cytokines and things like that. That gives you clarity on the difference between a multifaceted infection and single cytokines. And some of that work has been done in mouse models as well, where they've done all this initial primary work with just cytokine injections. And then when they do an actual, viral infection, it's very clear how different that is in terms of modeling. We now have a mouse model of CMV infection, but that's actually, when we model that infection, we do it immediately postnatally. We don't do it as a congenital infection because you actually don't, for one, you don't get vertical transmission with a CMV infection. That's not so much of a problem. It's just the survival aspect of congenital infections in mouse is really challenging. I'm not sure I answered your question.
No, that was great. Really cool talk, by the way. I enjoyed that a lot.
Oh, thank you so much.
Let's see, anybody else?
Hey, Anna, this is Hitesh. I'm sorry, my microphone was muted before I couldn't answer the question. So great talk.
Yeah.
So as you know, so Rich Loxley makes this case about setting up the immune tone, and that there is this balance between Th1, Th2, and Th17
Right.
environment that's set up in early life. And in the data that you have presented so far, it's just looking at skewing of Th2 responses. Have you looked at Th17 producing cells? So for instance, ILC3s and Th1 producing cells, ILC1s, and see how fetal inflammation is affecting the relative balance between these three cells. And also, can you comment on have you looked at long-term persistence of ILC2s and how that may be disrupted by early life inflammation?
Yeah, so I for the first part, I have not specifically looked at balance or output of ILC1s or Th1 versus Th17. We were specifically focused on the ILC2 itself and looking at its function and output. In terms of whether it affects long-term persistence of the ILC2, it does, because we see that ILC2 compartment is expanded, and we're still tracking those, but it certainly remains expanded. It is not as expanded as it is at P14, so it still contracts. And I don't know, I don't think it's terribly well studied how, I think that's probably niche regulated, and because you get input also, from Th17 cells and so forth. But we know that that the ILC2s are still expanded out. We've gone out to 8 to 10 weeks so far. So we don't quite yet know what the function of that expansion, what that means functionally, but we know that there's still more of them following prenatal inflammation than the saline controls.
Great, thank you. Any other questions? Okay, with that, we'll thank Anna again for a wonderful talk and presentation. It really evokes a lot of questions about, prenatal conditions for development, especially of the immune system. Really a great talk and great presentation. So thank you again. And thanks to all the participants. And take care, everybody.
Thank you. Bye.
Bye.